You know, one of the things I love about living in Switzerland? The post. Day in, day out, letters and parcels just arrive. No drama, no delay. And that got me thinking. Our bodies are kind of like that too. We've got about 37 trillion cells, each one like its own tiny city, alive with motion and full of activity. Inside every one of those cells is a bustling molecular post office, hard at work packaging and shipping out crucial cargo. That's where the escort machinery comes in, short for endosomal sorting complex required for transport. Here's how it works. The machinery reads molecular addresses, usually small tags like ubiquitin, so it knows where proteins go where. Then it bends flat membranes into tight little vesicles to ship them off. That process requires incredible coordination, like folding up a box and sealing it, but at the scale of billionths of a meter. Now, when this system goes wrong, chaos erupts. HIV, for instance, hijacks the escort machinery to package itself and spread throughout the body. It turns the postal system into a smuggling operation, slipping through immune defenses. So to fix these breakdowns, we need to understand exactly how this machinery works, from the ground up. That's the challenge, because inside a cell, everything is happening all at once. It's chaotic, it's tiny, it's fast. Processes unfold in seconds, most of the time less. So how do you study something so small, so quick, and so complex? That's what my PhD is all about, capturing membrane bending by escorts step by step. To crack this open, I used two complementary strategies, a bottom-up approach and a top-down one. Let's start with bottom-up. In this approach, I strip things down to their essentials. I purify individual escort proteins and add them to artificial membranes, clean, controlled setups. This lets me tweak the timing, control conditions, and really see what each component is doing. Using electron microscopy, I could see membrane bending and cargo sorting, capturing the process in the act down to the nanometer. On the flip side, my top-down approach dives into the real thing, living cells. I use yeast as a model system because yeast cells have escort proteins that are remarkably like ours, and they could be easily genetically modified. I knock out specific proteins and watch what happens. With live cell imaging, I follow the process as it unfolds in real time. Then, with electron microscopy, I zoom all the way in and capture the structural changes. To quote Richard Feynman, "What I cannot create, I do not understand." And that's my goal, to create the molecular post office from scratch. By doing this, I hope we can not only understand how cargo gets sorted at the cellular level, but also what happens when the system gets hijacked or falls apart, giving us the opportunity to intervene, repair, and even outsmart nature when it goes rogue.